Okay, so I'm Vir Gogoi, and my project was on the identification of potential biomarkers predictive of chemotherapy response in ovarian cancer. Okay, so our main objective was to determine the transcriptomic differences between poor and excellent responders in ovarian cancer, as well as being able to identify genes that were predictive of response. Why are we doing this? We're doing this because for breast cancer, the survival percentage is around 90%, while for ovarian cancer, it's only around 50%, or the 5-year survival percentage. So our two main challenges are late diagnosis and then recurrence and subsequent resistance. We have two ways of classifying response. First is recurrence duration. If the patient recurs in less than 6 months, it's classified as early recurrence, and if it's greater than 6 months, it's late recurrence. The other method is RECIST classification. So for RECIST classification, you have a pre-treatment and a post-treatment tumor sample. For complete response, it's the disappearance of all the tumor, the complete disappearance of the tumor. For partial response, it's a significant decrease in the tumor. Progressive disease is a significant increase in the tumor, and stable disease is a non-significant increase or decrease in tumor size. I'm going to get into a little bit more about the problem of recurrence and chemo resistance. So you have an untreated tumor. When you do chemotherapy, one of two things can happen. Either it's already resistant to chemotherapy, in which case nothing happens when you perform chemotherapy, or it initially is sensitive to chemotherapy, in which case most of the tumor will disappear. Eventually, though, it'll grow back, and after multiple relapses, or maybe not, eventually it'll acquire resistance. So either way, eventually the tumor will become chemo-resistant. Our first question, or our main question, was what are the genes that control poor or can indicate poor response to chemotherapy? Our hypothesis was that ovarian cancer that has poor response to chemotherapy has a different gene expression pre-chemo compared to the tumors that respond excellently. Our design, so first we obtained RNA-seq data from Tempest Lens cohort. All the patients that we got had high-grade serous ovarian cancer. They were all pre-chemo samples, and they all had platinum and taxane combination chemotherapy. The response we used was the RECIST criteria. For the bioinformatic work, we used RStudio, and then we used multiple packages like EDGAR, PCA tools, Mixomics, and Tidy models. The cohort we got was 112 pre-chemo patient samples. 70 of those were complete response, and 42 were poor response. The way that the samples were gathered, so first there's a pre-chemo biopsy. From that biopsy, you get a tumor sample. The tumor sample is then prepared and put on a slide. The slide is examined by a pathologist who determines if the tumor is large enough to do RNA extraction and sequencing on. If it is, then you do RNA extraction and sequencing. Using that data, we can perform DGE analysis. And with the DGE analysis, using the differentially expressed genes, we perform machine learning. Our first question was, what are the differences in RNA between poor and excellent responders pre-chemotherapy? We used patients from complete response and poor response, and we found the differentially expressed genes between these two groups. This is a graph, a volcano plot that shows the differentially expressed genes. We used poor response versus complete response, using complete response as a reference. As you can see, there are 186 upregulated genes, as opposed to the only 43 downregulated genes. With the DGE, with the differentially expressed genes, we performed a GSEA analysis. We did this to test the enrichment of positive or negative regulation of 10 biological processes, which we thought would change between the two different groups. So we did this on cell cycle, apoptotic signaling, epithelial to mesenchymal transition, immune response, lipid metabolic process, and stem cell processes. Out of all of these 10, we only found one which was significant, which this was the positive regulation of cell cycle. So this is the graph which shows that. As you can see, it is upregulated in excellent response. So yeah, that's the summary for the first part was we found that pre-chemo, poor and complete responders have differences in their gene expression. Most of the differences are upregulated genes in poor responders. The most upregulated gene is NCCRP1, which is associated with cervical cancer, and the most downregulated gene is GAGE12H, which is associated with testicular cancer. Complete responders have an upregulation of the positive regulation of cell cycle pre-chemotherapy compared to poor responders. Our second question was, can we use the RNA-seq data pre-chemotherapy to predict likely response even before treatment? To do this, we used machine learning. We used two different algorithms, a decision tree and a random forest. So to split the data, we had same Tempest lens cohort. We split it into training samples and test samples. Okay, so I can't explain that. So we used a Pca plot because we wanted to see if we could find genes which would separate the two groups or if they would naturally separate. So we do good response versus poor response. The Pca plot, it doesn't take into account the two groups, and it sees if it can separate the points using different patterns. But as you can see here, it doesn't separate it. So we have to go to a Plsd plot, which takes into account the groups to try and separate the two responses. So for the Plsd plot, the x-axis uses the 400 genes which can separate the two groups the most, and then the y-axis uses the next 400 genes. So because we need some genes which can separate the two groups, so we extracted the x-axis and we used that to subset the so this is the 400, or this is some of the 400 genes, and the lines show how much it's able to differentiate between the two groups. So this is our decision tree that we did. So it takes the 400 genes which we've got from the PLSDA, and it ranks them based on importance. So as you can see, FAM98C is the most important gene. So it separates the responses based on that. And so from this, if it's greater than 2.8, then it goes to poor response, but that's still not pure enough because it has some complete responses in it. So it uses the second most important gene and it separates it again until it reaches significant purity. This is a graph of the most important genes for the prediction, for the decision tree. And then for the random forest, this is the graph for the most important genes for that. So since now we have the two models, we have to see which model we're going to use. So we make a rock area under the curve, or a rock curve, sorry. And as you can see here, as you can see here, the random forest model is much more accurate, which makes sense because a random forest is just multiple decision trees. So for the decision tree, you can see that there we go, okay. We can see that the accuracy and the rock area under the curve is way lower than for the random forest model. So we extracted the 5 most important genes from the random forest model as possible predictors for ovarian cancer response. We also found that poor and excellent responders have different gene expression profiles pre-chemo, and that excellent response has an upregulated positive regulation of cell cycle pre-chemo. Our future plans are to validate these biomarkers for the random forest model using the TCGA cohort and to perform immunohistochemistry on tumor samples from poor responders and excellent responders to see if these actually are biomarkers. I would like to thank Dr. Moore, my PI, my mentor, Nick, the Moore Lab for their help, and the Discovery to Cure project for this opportunity.